Welcome to our webinar this afternoon. Before we start, just some housekeeping. If you have any technical issues, please send a short message in the questions box on the right-hand side of your screen. During the webinar, RSSL would like to invite you to ask questions using the questions box, which is also on the right-hand side. And at the end of the webinar, time permitting, we will try to answer as many of these as possible. Any we do not get around to, we'll answer offline and share with you all, along with a link to the recording. Today's webinar is on what's lurking in the water, controlling pharmaceutical water systems. My name is Mark Igmedian, and I am the Biologic Sales Manager at RSSL. I've recently joined the commercial team here at RSSL and have over 10 years experience in the biopharmaceutical and clinical sector. My role is to support our clients that require a variety of bioanalytical and microbiological testing from complex bioassay development to environmental monitoring and cleaning validation projects. For those of you who are new to RSSL, who are we? RSSL is a global CRO supporting clients with a wide variety of analytical services. We have been around for over 35 years and are based exclusively in the Reading area, where we have 4 separate sites. For those of you who don't know where Reading is, we're about 40 minutes west of London and Heathrow. Our 4 sites accommodate over 350 scientists working and supporting clients in 12 different technical areas, of which microbiology is just one of them. We support clients globally, with clients all around Europe, as well as the US and as far away as Singapore and Australia. RSSL is a trusted CRO with a big emphasis on quality, science, and service. This can be shown in our high Net Promoter Score. Because our site sits close to one another, we can also show great agility to manage complex multidisciplinary projects. We also have a dedicated product manager and client liaison managers to ensure those complex projects are kept on track and managed. The 12 technical areas we specialize in allow us to support a diverse range of biological, microbiological, chemical, and physical analytical services across all phases of clinical development through to commercial release. We are exceedingly well positioned to support your projects utilizing our state-of-the-art and experienced technical team who can advise and support you throughout your manufacturing. Be that routine raw material testing, investigative support of any issues you may face, such as impurities or foreign particles in your processes, we have a well-equipped and extensive microscopy department which can build a specific particle database for your manufacturing process. Our microbiological support is also extensive, where we can support all aspects of your Annex 1 compliance, including microbial identification of any environmental isolates or unfortunate positives for your bioburden analysis using our state-of-the-art Molitor system. We can also offer a range of turnaround times to ensure you get those IDs through as soon as possible to help support your decisions and next steps. Enough of me. I would like now to introduce you to our guest speaker, Dr. Tim Sandle. I'm sure many of you have heard of Tim and subscribed to his many publications. Dr. Tim Sandle has over 30 years experience in microbiological research and biopharmaceutical processing. Tim is a member of several editorial boards and has written over 900 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and also finds time as an associate professor at both the University of Manchester and UCL. Over to Dr. Tim Sandle.
Okay, thank you very much, Mark, for that introduction. And I will. Ready. Okay, the slide. Can you see the slides? Okay.
Yes.
Brilliant. Okay, well, welcome everybody. And there's a number of things we're going to be covering in this.
Sorry to interrupt, Tim. Your control panel's still up. You can just pop it away for me.
Yeah, that's weird.
There we go. Perfect. Thank you.
Hang on. Okay. Yeah, so we're going to be looking at what we know about microorganisms in water. We're going to be looking at some of the characteristics of water microorganisms. We'll be looking at some studies that have been conducted in relation to pharmaceutical water. We'll be looking at the factors that influence what we find, how we find it, when we find it. We'll also be considering what the limitations of the methods actually mean, so what we don't actually find, and we'll be looking at some of the implications. So we'll be touching on things like biofilms, endotoxin, filtration, Burkholder-Sapieha complex design, and so on. So there'll be some key topics that we'll be alluding to. But let's begin with a slightly philosophical question of what do we actually know? And the first thing is that because of the distribution of microorganisms in water, which do not conform to normal distribution, it's heavily skewed. Some refer to it as Poisson distribution. But essentially it means that we would need to take large numbers of samples in order to truly understand the nature of microbial distribution in water. And even when we are able to capture and cultivate microorganisms, then the counts do not really fall into homogeneous groups. So that means that our knowledge is limited, and that's a factor of distribution and the unevenness of recovery from water systems. And we can also divide the different types of aquatic bacteria that we recover from pharmaceutical grade water systems into either those that we can refer to as autochthonous, which is originating from the water system, and which might be the consequence of poor design. So, for example, if we have like a dead leg, a place where the water velocity significantly slows down. And we can also define some of the microorganisms we might discover as allochthonous, which is organisms introduced from a different environment. So this could be the consequence of poor maintenance or repairs. So, for example, a leak, or if an engineer conducts a valve replacement, for example. And then in terms of the importance of looking for and studying for water microorganisms, then conducting microbial identification is important because it helps us understand what is present within the water system. So it informs about the degree of control. It also helps us understand if there is any relationship between different grades of water. So where we're using potable water to create purified water, and then if we're using purified water to create water for injection, or if we're branching off to create a laboratory grade water, we can get a holistic overview of what may or may not be present. It's also important to compile data so we can understand the patterns of trending over multiple years, and that informs us about points of shutdown, maintenance periods, even certainly for early stages of water generation and purification, whether there is any impact of seasonality as well. And also trending allows us to see what is within the norm. So if we start seeing the detection of unusual or atypical species, this can inform about contamination risks as well within the water system. And we can also get a feel for whether there are emergent problems as well. And then depending upon what we're applying the pharmaceutical water to, we can also get an understanding as if there's any objectionable organisms that also might be arising. So if we're making a particular non-sterile product, so we're making an inhalation product, then if we were getting recoveries of organisms like Burkholderia cepacia complex, then that would be the type of objectionable organism that would trigger that level of concern. And there are other reasons why we would wish to characterize water microorganisms as well. Because by undertaking this level of profiling, we can take the most commonly occurring isolates and then use those to do things like go back and challenge parts of the microbial culture collection, hold example organisms for future reference. We can use representative isolates to challenge the very culture media that we're using to screen our water system, so part of growth promotion testing, media release. We may wish to test some of the general microorganisms that recover against disinfectants as well. And it also gives base information for conducting the longer term trends that I mentioned. So in terms of what we might find, there are some published reviews that are available, and these give an indication of some of the things that we might find. So I've quoted two reviews there on the screen, and I have the full references at the end of the presentation that you can take a look at. So according to McAllister and colleagues, then there is a predominance of organisms like Ralstonia pickettii and Brazorybium. And he and his colleagues were saying that the typical types of organisms that we will find are those that are adapted to oligotrophic conditions, that is very low nutrient, low carbon source conditions. Whereas Teresa and colleagues showed a very high predominance of organisms that are pseudomonads or of a part of the larger family of pseudomonad-like organisms. And then another study also showed this predominance of pseudomonads and also organisms that would come under Ralstonii, flavor bacteria, and Acinitabacter. And these are all gram-negative organisms, and they're the types of gram-negatives that are classified as non-fermenting bacteria, which means that they don't utilize glucose. So with gram-negatives in sort of a crude sense, we can divide those up into non-fermenters, which generally have an association with water, soil, environmental conditions, and the fermenters, which would include groups of organisms like the Enterobacteriaceae, and these are often the organisms that reside within the human body. It's not always exact as that, but it's a good kind of ballpark definition. It's also interesting about these water organisms that if antimicrobial treatments are a concern, then they will show different resistance patterns. Taking another study, this is my own study that I undertook over a 10-year period, and this was looking at mains or potable water, and this is water that's intended to be of drinking water grade. It's supplied chlorinated, but a pharmaceutical plant would need to remove the chlorine and to then soften the water through demineralization in order to get it to a state for the final part of purification. And then purified water is water that's produced, in this case, by reverse osmosis. So this is a semi-permeable membrane where the water is passed through at a substantial pressure differential, which enables the water to pass through and traps impurities and microorganisms. And then there's water for injection, and this was a water for injection system produced by distillation. Of course, we can alternatively use a two-stage reverse osmosis process, but generally distillation is a more robust process in my opinion, and this is where we turn water from a liquid to a vapor and then back into a liquid, which separates out the water from not only from living bacteria but also from byproducts like endotoxin. And from this review, the greatest diversity of different organisms was perhaps understandably in the mains potable water. The diversity fell away quite substantially, but again, it's that kind of general pattern of Ralstonii, Bulkardiria, Pseudomonads, and Flavimonas as well. So what we've got is consistency across 4 different studies. And remember, I'll give the references at the end, but there are some commonalities of patterns that are quite clear. So if we look at the mains potable water, then the consensus of these studies is that Pseudomonas fluorescens is relatively common. This is an organism that can grow at ambient as well as lower temperatures, and it has a particular association with agriculture. So this may suggest runoffs into rivers and streams, which are then entering into municipal water supplies. The second most prevalent organism was Bramundamonas vesicularis, and this has an association of water in general, but also has a predominance for getting involved in biofilms, in municipal water supplies, has a link to slime in particular. And then the third most common from the consensus of the studies was Ralstonia piquetii, common to soil and also to water. But we can have these kind of expected organisms, and then we can have the organisms that we actually find when we start monitoring water systems. And different types of systems will recover different types of organisms. And we're going to get variances through the time at which we sample. So we're going to get different periods of use. We will get variations on how well maintained a particular system is and a consequence of its water treatment, and also the distribution network. We'll get variations depending on the size of sample that we take. So with say mains and potable water, then you'll find some facilities might just test a 1 ml sample, others will test a 100 ml sample. The sampling technique of the operator can be influential, plus the overall maintenance and hygiene of the outlet, and also then how often sampling is undertaken and the intervals between samples. It's also a consequence in terms of what we find will depend upon whether we opt to characterize everything or whether we are restricting our identifications to things that might be above predefined alert and action levels. So there's the impact of where we're setting those levels. But in general, the methods of recovery that we're deploying, certainly through conventional microbiological techniques, are going to be the heterotrophic plate count, So if we're using the heterotrophic plate count, we will recover those water organisms that can be classed as heterotrophs, which means that they need an external source of carbon for growth. So there needs to be a minimal level of carbon that they can utilize from the water system. And the culture medium that is prescribed in the British and European pharmacopoeias is R2A. That isn't necessarily the case in the United States pharmacopoeia, where there's a kind of broader choice of different culture media that can be looked at. But R2A was a medium that was introduced in 1985 by the researchers Reasoner and Geldrich. And the R stands for Reasoners, and 2A is the second generation of the agar that he was formulating. And it's a medium that has far lower levels of nutrients compared to, say, tryptone soya agar. And this allows us to detect a higher proportion of organisms than are used to environments with lower nutrient levels. So it's basically the concept that if organisms are acclimatized to nutritively depleted conditions, then they're more likely to grow on growth medium that more closely matches those conditions. Research suggests that R2A yields its highest counts when it's incubated for 5 to 7 days, generally between 20 and 28 degrees Celsius when compared to tryptone soya agar. So you might ask the question, why does the pharmacopoeia talk about 30 to 35 degrees? That's a question I've never got a clear answer about, because the literature does suggest a lower temperature. Other influencing factors are the method of subculturing. So there are some microbiologists who say that if we're going to subculture from R2A for, say, microbial identification, then it's preferable to use an interim nutrient media like R3A as the bridging step, rather than trying to go straight from R2A onto TSA, because sometimes the shock is too much and the organism enters a non-viable state. And also influential is the database used. In previous webinars we've spoken about microbial identification systems and the fact that they always look for probability, and we can get variances from databases as the systems try to find the best matches. Another influencing factor is geography, because where we are in the world is going to affect the types of organisms that we find, and also it's going to affect the supply of water, and this might relate to local drinking water standards. It's also going to reflect the source of the water as well. So if our source water is coming from nutrient poor upland rivers, that will give lower microbial counts, but probably relatively hardy organisms in terms of their acclimatization. Whereas if we're drawing from lowland rivers, then these are sort of more eutrophic environments, and we're going to get far higher counts, but perhaps organisms that are then less acclimatized as we start to purify the water itself. Another thing is that incoming water supplies are going to vary relatively considerably as we get seasonal distribution as well, which is going to be a product of nutrient levels and temperatures as well. And we're also going to get variations in the amount of carbon that is available as well. And the best way to assess carbon is to look for the assimilable organic carbon content, which is a finer method of assessing carbon that organisms can actually utilize compared to the more traditional total organic carbon approach. We're also going to get influences from different methods of purification. And with any study, if we're going to go back and compare to studies, we also want to know what the historical window was as well, and how much that influences anything. But what we can infer to kind of bring this section together is that we're mainly going to see gram-negative bacteria. They're going to be non-fermenters. There will be a reasonably high level of taxonomic biodiversity, leaning towards Pseudomonas flavobacteria and Ralstonia. We will get very few gram-positive bacteria, and we will see we may see some gram-positive bacteria in mains water, but we wouldn't really expect to see any in purified water and none in water for injection, unless we were dealing with something very specific around a sample outlet that had become poorly maintained or contaminated. Another thing about the organisms is that they will also tend to be relatively small. And that's because the typical size of a bacterial cell, if you look in the textbook, you'll get an approximate range, will be substantially smaller as the nutrient conditions decrease. And also there will be sort of other influencing factors from any water system, depending on its specific microenvironment. So there will also be organisms that are present in water that we cannot recover. And this is either because the cultural conditions are not appropriate, either in terms of the media formulation or the temperature. There's also going to be some organisms that are simply too fastidious, with their specific physicochemical properties that are required. And there will also be organisms that might be readily recoverable if they were from elsewhere, or we were trying to grow them on standard healthy laboratory media, but the particular conditions, maybe patterns of osmolarity, low nutrients and so on, that they enter into a viable but non-culturable state. And gram-negatives are far more adaptable for entering the VBNC state than other organisms. It also stands that in a water system, we're either going to have a mix of free-floating organisms, or we might have some organisms that are attached to surfaces. And the attachment to surfaces is something that can sort of lead us towards biofilm. So from standard water sampling, we will get a detection, a bias in our detection towards those organisms that are free-floating, or what's called the planktonic state, because it's quite hard to detect unless they are shed or eluted off those that are attached to a substratum, or part of a biofilm community. And we can often know there are higher numbers than conventional methods can show by applying methods like flow cytometry, which often show higher cell numbers than any cultural method can show. And we can also gain an idea of sort of greater species richness by applying methods like 16S or RNA gene applicant analysis, which just reveals these kind of richer, deeper patterns. But recovering microorganisms is not necessarily synonymous with survival. Whether organisms survive long term in a water system very much depends upon whether or not they have the opportunity to become attached to a surface. So it's the whole act of logging onto a surface that boosts their survivability. So survival really needs a substrate for surface attachment, and one that's suitable. A level of nutrients, and this can be incredibly low level, but there needs to be a level of nutrients and a carbon source. And there also needs to be an absence of very high shear forces, which is a consequence of very, very fast water flows. So the conditions that can lead to attachment and the conditions that can help to protect the microorganisms are often related. So with a water system, we want to have very, very fast moving water. And we express that through a dimensionless unit called the Reynolds number. And we want very, very rapid turbulent flow, generally exceeding 4,000 on this scale. So when we start to slip below that, we start moving more towards laminar flow. And in water, this is the kind of flow we don't want, because one, it can enable organisms to attach to surfaces, and also avoids excessive shear forces, which can be a counter effect to biofilm, so that disappears and makes the biofilm more likely. Okay, so we've got a bit about the types of organisms, we've got a bit about the variable patterns of recovery, we've also got some of the factors that help to promote survival. But what can we make of this? What lessons can we learn? Well, the first thing we have to be mindful of is that the thing we're seeking most to avoid is the biofilm. Because once a biofilm takes effect, once it moves through a level of maturity, then it becomes harder and harder to get rid of. And we'll look at ways to get rid of biofilms shortly. But in terms of the biofilm, is that it stands that many of the organisms that have adapted to survive in low nutrient conditions are also the types that are most adept at surviving and forming biofilms. So Ralstonia beckettii, for example, is such an organism, and it also happens to be a common community member of where a biofilm might develop in a pipe together with pseudomonads. And once organisms become trapped within a biofilm, then what will happen is that the biofilm will develop a outer slime-like coating that will give it a degree of protection from water flow and from subsequent chemical treatment, and to an extent heat treatments. And also the biofilm will slowly release individual cells, or it might be greater clumps of cells, or it could be in the form of endotoxin. So it becomes a problem, and the rate of release is very skewedly distributed, which is why if there's a biofilm present, you can get kind of results of passing for a while, then the occasional out of specification, then a run of results passing, then the odd out of specification again. And it's not contamination coming or going per se, it's the rate of biofilm release that becomes a problem. I mentioned endotoxin. So high levels of gram-negative organisms are problematic in not only the organisms like to be present and the organisms most likely to form biofilms, but also in relation to endotoxin generation. And it's been estimated that as few as 2,500 cells of an organism like Burkholderia cepacia, or around 3,500 cells of an organism like Pseudomonas fluorescens are sufficient through the amount of lipopolysaccharide that they can theoretically release to give a detectable level of endotoxin that can arise with the allow test. So endotoxins can be shed from or viable bacteria. And the most common habitat for gram-negative is obviously water. And the typical amounts of endotoxin that are associated with a gram-negative bacterium are quite small, but they can come together and contribute to significant levels. So the average bacterium has 10 to the power of minus 15 grams of endotoxin. So that's obviously a very, very, very tiny amount. But the levels of endotoxin, the number of kind of cells that are required to trigger a level of endotoxin of around sort of 1 endotoxin unit are around 10,000 cells would be equivalent to around 1 endotoxin unit from a common pseudomonad type organism. Other things about endotoxin is that the type of environmental endotoxin produced by gram-negative bacterium water is highly heterogeneous. Potency will vary according to bacterial species and strain, but also by the formation of the endotoxin molecule as well. So in terms of its solubility and molecular weight. And the more potent endotoxins are those of the highest molecular weight, and those that are most disaggregated. Another thing about the kind of heterogeneous nature of endotoxin from water systems is that it tends to aggregate to form what's called vesicles or membranous structures. And the size of these vesicles is dependent upon the type of lipopolysaccharide, the structure, and also the environmental conditions of the water. So in terms of pH, salt concentration, and so on. And the size is also varied. So we can have endotoxin from sort of 20,000 to 100,000 Daltons. Another factor with endotoxin in water is that it is also relatively sticky, so there's a tendency to have an affinity towards surfaces. I also mentioned earlier that the sort of relative size of these organisms is quite small. So we do have problems here of organisms becoming so small that they could theoretically pass through a sterilizing grade filter. So we have 0.22 micron as a sterilizing grade filter, and organisms that are normally too large can become so small or to a degree plebomorphic in their structure, or kind of like the degree of their plasticity, that they could theoretically pass through filters of this type. So if we were to get high levels of certain organisms, then we do have a risk to our filters from those. There are also certain organisms that are classed as objectionable. And depending upon the products that we're seeking to manufacture, there are certain organisms that pose direct risk to patients. In the US, and we can see from FDA guidances, there's been a lot of publicity around the Burkholderia cepacia complex, of which there's 18 different organisms, bivalves within this group. And these are organisms that are especially equipped for survival of low nutrient moist environments. The problem is that these organisms are human pathogens, and they can form pneumonia in particularly in immunocompromised individuals. So certain inhalation products can be at particular risk from this organism, particularly in purified water systems. So the FDA has put out various warnings to manufacturers of inhalation products about this complex of organisms. And this has required manufacturers to considerably improve the control of their purified water systems and to put emphasis upon sanitary design, equipment cleaning, environmental monitoring, and so on. And the United States Pharmacopoeia has developed a specialized culture medium that requires an enrichment step for the cultivation of this organism. Understanding the kind of nature of organisms in water also places an emphasis upon good design. So we can overcome many of the challenges posed by aquatic microorganisms through good water system design. And perhaps the most important factor is having rapid turbulent flow, which minimizes the opportunities for colonization. So that's all about fast flow, high water pressure, suitable pipe material. So organisms are less likely to attach to stainless steel than they are to plastic piping, having smooth surface finishes, absence of dead legs, and being hygienic valves as well. So these are all key factors that come in to the equation. Another thing about water is the nature of water activity. So again, for non-sterile products, whether a waterborne organism is likely to survive within a product and pose a risk to a patient depends on the level of water activity of that particular drug product as well. And this is not about how much water, but the degree of free water that is available for a microorganism to use for a biological process. So the definition of water activity is one of partial vapor pressure of water in a substance. And this can be affected by temperature, water purity, humidity, and the nature of the substance itself. And water activity is measured on a scale of 0 to 1. And the higher the water activity a substance, then the more likelihood it is that the substance will support the growth of microorganisms. And generally, when we get to 0.91 and above, we have sufficient water activity to support bacterial growth. Fungal growth is something different. A lot of fungi are capable of going down to about 0.7. But generally for risks from bacteria, we're going from 0.91 upwards, and for gram negatives in particular, we're looking around the sort of 0.96, 97 mark. Regular monitoring, as I've mentioned, is important both for viable counts and for bacterial endotoxin. And Annex 1 places strong emphasis on regular water monitoring, particularly in relation to water generation. And if we're running a facility that's operating 24/7, then we need to understand the quality of the water maintained over different points of use, and to ensure that we're conducting good out of limits investigations when we get excursions. And trending, something I also mentioned earlier, the trending of results from a water system is also really important. It helps us to infer the trend and to see patterns in the data. And the only real clear way to show trends from a water system is conducting charts. So we're looking at variations in data. We're considering how variations change with time. We can gain an understanding of which variations are expected and which are not. And also trying to divide up what might be common causes and what might be special causes from a water system. So special causes are often localized events. So this could be a problem with a water outlet. It could be a new person who begins sampling. It might be local tubing management. It might be something to do with laboratory testing, etc. Whereas common causes might relate to factors that are affecting the whole system widely. It might perhaps like the sanitization frequency or something like that. With trend analysis, it's an example of just a trend on the screen there. So this is looking at the overall counts from a water system, and you can see there's a clear case of a trend going up, and you would hope ideally that that rising trend and the reasons for that rising trend will be captured relatively early. And it's about kind of understanding, what are the factors that could be affecting such a trend. So is it one problem outlet? Is it a series of interrelated outlets from the same loop? Is it something to do with the generation plant? Is this following a maintenance activity? Was the system suitably sanitized following maintenance? Was there a loss of power, loss of pressure, a loss of water flow? Was there an equipment malfunction, say with pumps? Did the chemical or heat treatment not work correctly? Is it that our overall sort of periodic sanitization frequencies are insufficient for achieving the level of control that we are seeking to achieve? So that's the kind of main part of the presentation completed. So some of the references I've referred to are sort of listed on the screen there, which you can look up at your leisure. And then just to summarize what we've looked at. So trying to look at kind of what we know and why we know what we know, which is limited to an extent by different forms of culture media and the different physiological states that some organisms will enter into, and the variances of microbial distribution within water, which can affect what we find. Then we've looked at some of the characteristics of water microorganisms, the most common types, how they undergo size changes, how they adapt to different nutrient conditions, and whether or not there's a tendency to form a biofilm, which then becomes something more problematic. And also with biofilms, we've also got problems with endotoxin. And then there are various factors we need to introduce when we consider risk overall. And the example I used there was from an example of an ejectionable organism or group of organisms, as with the Burkholderia cepacia complex, and also with water activity from products, and how different products are going to be at a bigger or greater risk depending upon whether they promote survival or not. So anyway, that's the main part of the presentation completed, and happy to hand you back to Mark, and we can see if there's any questions.
Thank you very much, Tim. Thank you very much for that information and that presentation. We have had a number of questions come in, and so in the time remaining, I'd like to see if we can kind of get through some of these. So the first one I've got to, is it a risk that R. piquetti can pass through a sterile filter at 0.22 microns?
Yes, it can be. I think if the organism is found in abundance, then that could be a theoretical risk, but it's sort of kind of in sort of context. So I mean, how much is there, what degree is it in the water, is that water then used to formulate a product, is that organism then likely to survive in the product formulation? And if all those answers yes, then of course you could have a risk to this sterilizing grade filter, but there's a few sort of ifs and buts that can lead us to in that direction.
Okay, great. Thank you very much. The next question we've got is it common to observe fluctuations in results from the same sampling point when tested daily?
It can be, but it sort of depends on what the variables are. So you know, it might be something like sampling technique. It might be something to do with the control of water sampling tubing. It might be something to do with sort of splashback of the sink. But if all those things start to be ruled out and there are sort of fluctuating counts, then it could be something else that is amiss with the system. So it might be water that's been retained within a valve. It could be a section of pipe work that's not seeing sufficient heat or chemical treatment. So for a fully working, well-maintained system, there shouldn't really be variation, but there are a lot of different factors that could lead to such a variation.
Thank you very much. And then we have, how can we measure the AW value?
There is a specialist equipment that would do that, that will look at the water vapor pressure, or you can probably more easily is send samples of the product away and get the water activity levels assessed. And then there's plentiful of literature to sort of look at the AW or water activity profiles of different organisms, and to work out whether or not they are likely to survive, or whether they're posing a particular risk to a particular product family.
Okay, great. Thank you very much. Let's see if we've got time for one more. Let's have a quick. Do you see any signs of seasonality with the data?
I think with the sort of drinking water supply and mains water, then there is a sign of seasonality. Definitely it fluctuates, often being higher in sort of spring and summer, and then dropping down in the winter. For a well-maintained pharmaceutical system, once you've kind of passed the point of purifying in terms of softening and filtering, it would be less likely unless there's something that's gone awry with the water system overall.
Okay, I think we're close to the edge of time. So just to say thank you very much, Tim.
Thank you very much.
And thank you everyone for attending our webinar. I hope you have found that useful, and we appreciate the questions that you've asked. Any questions we didn't cover during the Q&A session, I'll forward to Tim. And as mentioned at the start, we'll circulate these along with the recording. If you wish to catch any of our previous webinars or Tim's blog or regulatory update, please go to our website or keep an eye on our social media posts. We also offer a range of technical training courses designed for professionals across the industry. We've got several upcoming sessions, including those detailed on the screen, with more scheduled throughout the year. You can find full details and registration links on our website. Remember, also check our website for future webinars. Just finally say thank you very much and have a good day.
Thank you.